Alright guys, so I just wanted to take a minute just to go over the cell cycle lab that you're going to be doing today. It's actually pretty similar to what you did freshman year, just it's amped up just a tad. And so you're going to be getting your lab handout that looks like this, okay, and so this is what I'm looking at as I'm doing this video. I'm sorry about the weird camera, but I have two screens. So I just kind of wanted to explain exactly what you're going to be doing. And mostly you're going to be counting cells today. And so this is an example of what you're going to see in the lab. So the microscopes are already set up and focused for you. And so the only thing that you should have to do on the microscope, if anything, is you can adjust the fine adjustment knob, which is the small knob that helps you focus. You really shouldn't be messing with the coarse adjustment knob. You could break the slides if you do that. But what you're going to do is you're going to go to a station, and at that station you have a microscope that is already focused, and you're going to have a field of view. Now, I believe most of your slides look like this. They kind of have this stain to them. They're about as clear as you can get them. They're magnified about 400 times, and so they're going to look very similar to what you see here. And so what you're going to do is, as a group, you've got to count 3 fields of view. So just to kind of save you time, that's why the microscopes are already set up. Now we need to make sure that we know what we're looking for, and that's really the key. You have a picture of what to look for at the station. You also have some pictures in your note packet. And so I want you to kind of maybe take a look at this page here. This is the one that I think is really going to help you the most in terms of identifying. And once you go to start counting cells, you're just going to have to be confident in what you know, okay? You're just going to have to go for it, and there is no right or wrong answer. I mean, any field of view is going to have a different number of each stage, and so you just have to do your best to identify them to your best ability. So the biggest thing is we need to know what we're looking for, okay? Now what I like about this particular paper here is it actually shows you that the stages can look different, because keep in mind that this cell cycle is ongoing. And so I mainly want to talk about what are you looking for, and then how are you going to go about counting your cells. So let's start with interphase. I do want to preface by saying that the lab is titled mitosis, but technically it should be cell cycle because we are including interphase, and interphase is not part of mitosis. And so in interphase, the cell is resting, growing, doing normal cell things, and the nucleus is still intact. And so for me personally, what I kind of look for an interphase cell is I look for the chocolate chip cookie, okay? I look for a complete nucleus, and in that nucleus, you're going to have nucleoli, and so those nucleoli kind of look like chocolate chips. So this right here, let's see if I can... this right here would be an interphase cell. Okay, this would be an interphase cell, interphase cell. What you're looking for is a complete intact nucleus, because this is when the cell is doing normal cell stuff. The DNA is in the chromatin form, and so it's not really visible. And so you should see a complete outer boundary as well as the nucleoli. And if you remember, in the cell cycle, the majority of the cell cycle is spent in interphase. So it would make sense that you're going to have a lot of eyes, okay? Now in prophase, this is actually the longest phase of mitosis. It's not the longest phase of the cell cycle, but it's the longest phase of mitosis. And there's actually kind of 3 key events that are going on in prophase. Remember that the nucleus is breaking down. This is the point at which we get the chromosomes, and the spindle is starting to form. Now, the spindle is not really relevant in the lab because we cannot see the spindle in terms of microscope pictures. And so what we're really going to be focusing on is a nucleus that is breaking down, and we're looking for chunky chromosomes. And so what I like to look for a prophase cell is a chunky broken nucleus, kind of like this one here. All right, so here you had an interphase, it's complete, it's intact, whereas here you can see that outer boundary is no longer solid, and it's starting to kind of look noodly a little bit, and that's because we're getting the chunky chromosomes. Over here, this is another prophase, there's another prophase, there's another prophase. So it still has that circular shape, but you can see the chromosomes, the nucleoli are also pretty much gone. Remember that they're breaking down along with the nuclear envelope. And so on this paper here, what I wanted to show you is that you actually have 3 different prophases on this particular image because they're showing you an early, a middle, and a late prophase. And so just kind of in each one you can see that that nucleus is breaking down, and you can kind of see those chunkier chromosomes. So you're looking for a chunky broken nucleus, okay? All right, now the other ones are pretty easy to identify because they are very distinct. And so remember that in metaphase, the chromosomes line up in the middle. So you should see a linear formation. Okay, this is going to be a metaphase here. If I'm looking at some of these other slides, like that's a very clear metaphase. That would be a metaphase there. In anaphase, the chromosomes are being pulled apart. This right here is an excellent anaphase. But once again, you're going to have an early and a late. So there's going to be an early anaphase where they're starting to pull apart, and there will be a later anaphase where they're further apart like you see here, okay? So you just kind of have to be the judge, because remember, it's ongoing. So that metaphase is going to fade into early anaphase, and it's going to keep going. So what I typically look for anaphase are claws. It looks like there are two claws like here, and you can definitely see the space in the middle. Now, because we're dealing with plant cells, when they are undergoing cytokinesis, they have the formation of a cell plate. And so like in this one here, in this cell here, you can see that they're on opposite ends, but they're actually starting to go back into circular formation to form a nucleus. And you can also see this faint line right here. And that right there is your cell plate. And so the cytokinesis stage is not part of mitosis, but it starts happening around telophase. And so when you see a cell like this, you can see two new nuclei, and then you can also see that cell plate. So if that cell plate is visible, then you have a telophase, or a cell that's in telophase. Over here, this is also kind of probably a telophase because they are completely on the opposite sides, and we're getting back our two new nuclei. It's very faint, but I think that cell plate is forming there. Over here, like this one here, this would actually be two cells in interphase because these are two cells that were recently made. You can see that they're smaller than the rest, but that line goes all the way across. So that would be your new cell wall. And so if you're trying to distinguish, is it a telophase or are they back in interphase, look to see if that cell wall is complete, okay? Down here would be another telophase. And so what you're looking for telophase are two new nuclei. They're normally going to be smaller because we've divided the volume of the cell in half. There's another telophase there. You can see the cell plate very clearly. And so essentially what you need to do is just know, what am I looking for? So for interphase, you're looking for a complete nucleus with nucleoli in it, the chocolate chip cookie. With prophase, you're looking for the broken nucleus. It's got the chunky chromosomes. Metaphase middle, anaphase are going to be the claws because they're being pulled apart. And then in telophase, you should see two new nuclei, and you will probably also most likely see that cell plate, which is occurring during cytokinesis. Now the lab itself, pretty much what you're doing is you're counting cells, okay? And I would really like for each person to at least do one field of view, okay? So ideally you'll be in a group of 3, and each person will do a field of view. So if you look at your data table here on the back, this is what you're going to be filling out. What you really need though is a piece of scratch paper because you're going to be doing lots of tally marking. Now what I would not recommend doing is trying to look at a field of view like what you see here and trying to count interphase and then trying to count prophase. Ideally you want to hit every cell in that field of view that you can. Now sometimes there are cells like here, there's not really even a nucleus. Like these, they're completely blank. We're not going to count those. Only count cells that you can clearly identify a phase in, okay? Now, what I would recommend, and I know this can be hard on the eyes, and so you may need to take a break, but what I would recommend is if this is my field of view, now keep in mind it is a circle, okay, not a rectangle, I would kind of maybe start in the top left, and I would either, kind of go down and back up. And what you need to do as the person who's looking in the microscope is you need to be calling out the phases to the members in your group. Someone needs to be tally marking. So I would get a scratch piece of paper and I would write I, PMAT. And so what you're going to do if you're looking in the field of view is you're going to start up here and you're going to say, okay, I. And you're just going to go cell by cell and you're going to identify that phase. It doesn't really matter where you start or where you finish. It doesn't matter what pattern you take, but you want to try to count every cell in that field of view. So most likely you're probably going to have anywhere from 75 to 200 cells depending on that particular field of view. And you need to do that 3 times. So I really want each student to do one field of view at least, okay? Now, the reason the microscopes are already set up for you is to save you time. And also, I don't want you using the same microscope for multiple fields of view. So instead of you moving the slide, what you're going to do is you're going to go to another station. And that way you have for sure looked at 3 different fields of view. If you're using the same slide and you're just moving it, you could easily double count cells. So you're going to go to a station, you're going to count every cell in the picture, just kind of do your tally marking on a piece of scratch paper. You're going to go to another station, do the same thing. You're going to go to another station and do the same thing. So you're going to do that 3 times in order to fill out your data table, okay? Now, once you fill out your data table, there's a column where it says the total number of cells, all right, and that's where you're going to add up how many cells were in interphase, how many cells were in prophase, in each of the phases, okay? Then it has a time in minutes, and so if you look up above in this section of your lab, there's a formula for how to calculate time in minutes. They're telling you to assume that this whole cell cycle is taking 720 minutes. So you're essentially doing a ratio, okay, the number of cells in that phase divided by the total times 720. And that's going to tell you how many minutes that cell was spent in that particular phase. And then you're going to do the same thing and you're going to do just a percentage. Now to do the percentage, you can do it one of 2 ways. You can do total number of cells in that phase divided by the total of cells times 100, or you could do the minutes, the time in minutes divided by 720 times 100. And either one of those will give you the percentage. All right, now the last thing we need to talk about is your kind of what you're doing with this information. So you don't really have a lot of questions to answer or anything like that. All you have to do is a graph. And so if you look at on your handout, number 12, it says prepare a bar graph. Okay, so it's telling you what kind of graph to use. It says your independent variable is going to be the 5 stages of the cell cycle. I did change the term mitosis because technically it is the cell cycle. We're including interphase as well as PMAT. And your dependent variable is going to be the time in minutes. So you're going to be using the time column. You're not graphing the percent, you're graphing the time. And I just want to remind you to, as always, anytime you're making a graph, you need to include a title that represents what that data is telling you. You need to label your x-axis with units if necessary, y-axis with units. You need to have even increments, even spacing, and you should take up a minimum of 3/4 of the graph in each direction. Okay, so in terms of grading, this is not a difficult activity, but you need to have all the elements of a good graph. And so at this point, you can get in your groups, you can start counting cells. If you have any questions, Ms. Petty is there to help you, and I hope you guys have a great day. I do plan on coming up there on Friday, and hopefully if you have any questions or anything, I can help you with those on Friday. All right, y'all have a good one.